Alright, so let's explain a few things. We're going to explain DNA synthesis and protein synthesis, which is a form of or not tRNA, it's going to be messenger RNA. So we'll get into a little bit of transcription and translation, and then we'll get into a little bit of the nucleic acids, a little bit of the structures behind the underlying biological mechanisms. Now, for me, I think the chemistry part is a little bit easier because everything um, can kind of explain itself if you know a little bit of chemistry. But if you don't, it'll be a little bit more difficult to understand the conceptual ideas behind everything. For example, like we talk about hydrogen bonds, and um, you would have no idea what a hydrogen bond is if you hadn't taken like gen chemistry. So we'll get into everything first. So before we start, it's important to note that the two most popular nucleic acids are DNA and RNA, which are called information molecules because they're hereditary. They hold hereditary information. ATP, which is the energy molecule, is actually it's a nucleotide, which many people probably know that, uh, but it can also be surprising as well. So it's a nucleotide, meaning that um, it consists of a sugar, a nitrogenous base, and a phosphate group, which it's called adenosine triphosphate. So I think it has three phosphates attached. Uh, so yeah, the structure is I think it's five molecules long, or not five molecules long, five elements within its molecular structure. So a DNA is a library, it's genomic, and a gene is kind of like a recipe for a protein. So uh, in DNA, there's a strand. There's two strands, and what they call it is, I believe it's alpha helix or beta plated structure, and one has codes for one gene. So usually in textbooks, you'll see that this entire portion uh, will have a gene, or each strand, or this entire part codes for a gene. But actually, each strand just codes one, or not. Each strand, one gene is only encoded by one strand. It's not two at the same time. So that's important to know. So now we have to cover the differences between eukaryotic and prokaryotic cells. Those are pretty simple. Well, the thing they want you to know is about DNA storage. So, while the two obvious things are that prokaryotic cells don't have any membrane-bound organelles. They don't. The only thing they have is ribosomes, but ribosomes is not membrane-bound, and people will consider them bacteria due to, well, I guess it would be. I forgot the endosymbiotic theory or endosymbiosis, which is widely supported, by the way. But anyways, prokaryotic cells are bacteria. Eukaryotic cells are animal and plant cells that can do things like cellular respiration with the mitochondria, since that's an organelle. So for eukaryotic cells, I'll say ENP. Um, it looks like this, and let's just say that this is the nucleus. And again, the nucleus stores genetic information, so we'll see some chromatin in here. And right here for the oh, that's a P. Couldn't even read my own handwriting. Let's just say that this is the cell. The DNA is just everywhere. There's no way to protect it. And the reason why we need a nucleus here and we need the nuclear envelope to protect the genetic information is because there's something called I forgot what it was called. I think it was called nucleases. Um, Let me check real quick. Yeah, nucleases. Um, and they're basically just enzymes that break down nucleic acids and hydrolyze the bonds, and they're very important for immune defense and stuff. So we need a um, envelope for that. So we'll move on to other stuff. So the building block of DNA are called nucleotides. You should have known that from the macromolecules video. And nucleotides is composed of, um, well, I'll just write base, sugar, and phosphate. I believe, actually, these should be swapped around. Sugar, which is just anything that takes the form CH2ON. Base, which is just one of four. You have adenine, guanine, cytosine, and thymine. And you also have a fourth in RNA, but we'll get to that a little bit later. Phosphate, which is just PO4 minus 3, which you should know if you've taken regular chemistry, and a base AgCT and uracil, but we'll get to that a little bit later. So between these, they DNA has a deoxyribose base, which is just a different type of sugar. Oh, not different type of sugar, and RNA uses ribose. So you can remember that with the deoxy and whatever, and however you want to memorize it. But that is the logic there. So I don't know what's next to explain. Because my notes are kind of all over the place. Also know that proteins are bonded through peptide bonds, which are kind of logical. And a peptide bond creates polysaccharides, which is just essentially combinations of the thing. So, okay, so there's something called a sugar phosphate backbone, and that's pretty easy to understand. It's basically when DNA bonds together. Usually you'll see DNA drawn like this, as a alpha helix structure, and but like if you zoom in to the individual things, the individual, I forgot what they were called, like the rungs of the DNA. You'll see adenosine, thymine. Usually they'll be drawn like this, um, and guanine pairs with cytosine. Um, and you'll see on the side here that each is connected to a sugar, and on that sugar there will be two phosphate groups, usually. So that's what it looks like. And there's complementary base pairing, which just means A goes with T, G goes with C. And messenger RNA, A goes with U, T goes with, or A goes with U and T goes to A. We'll get to that a little bit later. So yeah, that's the logic here. Now, in double helixes, if you look at the structure, and I know my drawing is not the best, I'm gonna try to make it obvious. So like let's say it looks like that. This is what's called a major groove, and this is what's called a minor groove. A minor groove is something that's not too, there's not too much of a crazy bend. A major groove would be basically a larger angle. So yeah, this is a double helix, and yeah, it's essentially what happens there. So now we get to something called anti-parallel reading. So when you're reading DNA molecules, there is a 3 side and a 5 side. A 3 prime side, a 5 prime side. And in the same way, this is 5 prime and 3 prime. Or wait, 3 prime, 5 prime. Yeah. So you want to read it opposite. So if you're reading it this way, then you have to read 3 to 5 prime this way. You just read it the opposite direction. So if it's like A, C, G, then you'll just read it. Well, it would have to be complementary base pair, so it would be T, G, C, and you would have to read it up and down. So for example, if you have a 5 prime here, and the nitrogenous base structure is this, it's pretty simple to understand that DNA is read in an antiparallel reading. So 5 to 3. Wait, so then it would be 3 and 5. So it would just be read 5 to 3. Or you just swap the two numbers, which you can do as well, if that helps you memorize a little bit better. So yeah, a nucleotide is just composed of a sugar, phosphate, and base. Cell replication is mitosis. It starts with a diploid cell and splits into two identical cells. Meiosis, there's a G1. As I already explained all that. Prophase is where the chromosomes condense. Tetrads for meiosis 1, stuff like that. Simple stuff, so just memorize that. Uncontrolled cell growth is cancer, or uncontrolled cell growth, yeah, with mass is cancer. Know that too. For DNA structure, the base pairs weren't actually bonded to each other, and what I mean they're not actually bonded, they're not bonded covalently or ionically. They're bonded by electrostatic attractions called hydrogen bonds. All you have to know is the fact. You don't have to look into the molecular structure of it. These are just electrostatic bonds because everything in nature is just positive and negative charges, and they're just super close, and they're attracting each other because opposite charges attract. So that's the logic behind that. So Wow, there's a lot of notes. So I'm just gonna explain the basic idea, and the basic idea is called the general dogma of biology. And the general dogma of biology is just this: DNA goes to mRNA, which goes to protein. This is transcription, so I'll write trans C and translation, simply because this one letter differentiates the two. So it starts with the DNA, it gets ripped apart, and then a complementary mRNA comes on the side, bonds together base pairs. A goes with U, G goes to C, C goes to G, and T goes to A. So just find these bonds together. So now in DNA, what happens is it gets ripped apart, and one side of the DNA gets because it's just bonded hydrogen bondly. Also, the sugars and nitrogenous bases are bonded covalently, which are also called phosphodiester bonds, but that gets a little bit into the deeper chemistry behind it. So in transcription, essentially all the DNA from A, G, Z to T, all these nitrogenous bases get their complement. And this complement is kind of like a key, and this key is used in other areas to form the proteins. So the keys are called codons, and the codons are start with 3 and they end with 3. So there's something called stop codons and start codons. So with these 3 codons, they just there are certain codons for protons that tell it the ribosomes to start producing the sugar or the proteins. So, for example, there might be like CGU or something, and that might correspond to one of the amino acids. So maybe like leucine or something. I don't know. Um, I haven't memorized the countless stop codons. So leucine gets produced, and it forms this long polypeptide chain. And what a stop codon does is essentially a stop codon. There's 3 stop codons, I believe, traditionally, and it tells the ribosome to stop making it, and the ribosome stops making it. I think it gets split apart or something like that. But the stop codon essentially tells the ribosome to stop making the protein. So the first start codon that you have to memorize is called OG. And once the thing reads OG in that nitrogenous base pair, so like for example, let's just say it's C, T, U. Oh, actually, there's no T in RNA. G, whatever. It only starts when it reads AUG. So all these other ones aren't read. And I don't know the. I asked my biology professor. He doesn't even know the reason why this happens, but it does. So there's probably some explanation out there, but I unfortunately don't have it for you. So DNA starts with the genes. Transcription happens where it gets put to messenger RNA, and the messenger RNA is read by the ribosomes, and it's just it's threaded through each ribosome because there's hundreds of ribosomes, even thousands, I believe, and they just get read through and form the proteins. So that's the logic there. Mutations are just when there's variations within the nitrogenous base pairs. So like a traditional blood cell, for example, might be GC, ATCG, and a sickle cell one might be GCTTCG, and that small one changes it because the messenger RNA transcribes this as A, while here it would transcribe it as U, and that would change the stop codon entirely, such that like GUA would be read for the stop codon. Here it would be CAA or GAA, which completely changes the entire protein. So, or the peptide at least. And typically shorter peptides are mutations, so know that, and that's essentially all you have to know for the general dogma of biology. So